Today we're going to learn about the specific immune response. Before I go ahead and get started, this is the first time that you're watching a video of a lecture. So we're going to try this and see how it works. The great thing about this is you can pause at any time and stop and rewind and get any notes you need. The notes will also be on the OneNote just in case you miss something. Now back to the immune response system. So we're talking about the third line of defense, which is tailor-made to a specific pathogen. The specific defense system is specific, meaning that it responds to each unique pathogen. You have specific cells in your body that are designed to fight anything you could ever possibly encounter, including cells that are specifically designed to fight COVID-19. You just have to find the right cells in your body that can recognize that pathogen and then attack it. So the first step is really recognition. So step one, recognizing self from non-self. You got a lot of cells in your body. Some are yours, some are not. So how does your body keep track of all this? It keeps track with this thing called major histocompatibility complex, aka MHC. The major histocompatibility complex are proteins that are on your cells that your body makes to help you recognize self from non-self. The way it works essentially is every cell that you make gets a flag on it that says it belongs to you. So I'm Miss Shope. Every cell that my body makes gets an MHC that says Miss Shope, Miss Shope, Miss Shope. That's great. It's a great way for my body to recognize, oh, I made this. This is me versus this other thing that is not me. So what your body is looking for is your body is looking for a cell without your unique MHC on it, and it starts to attack it. You're probably wondering, wait a second, what about people who get transplants? And if you weren't, now you are. So people who get organ transplants, this is a problem. We try to type organs to match, and what we're trying to match is that major histocompatibility complex. The hope is that we can find a donor that has an MHC that's so similar to someone else that their body won't attack it. Still though, our body is really smart at knowing that's me, that's not me. So if it does see something that's not you and it's something you need, like for instance, a heart, it can sometimes get problematic, and people who have had transplants sometimes have to stop their immune system from attacking the organs that they need. But I digress. Let's go back to a normal pathogen invader and recognizing self from non-self. So once your body finds a cell that it doesn't recognize, that doesn't have your little MHC flags on it, then it finds cells that are specifically designed to fight that specific invader, and then it attacks that specific invader. This occurs through two different pathways, and both are kind of happening at the same time. You have your cell-mediated immunity response, which uses T cells, and you have your humoral immunity response, which uses B cells and antibodies. Now, T and B, humoral, cell-mediated, I have a hard time remembering this. So the way I try to think about this, cell-mediated sounds like there's a T in it, even though it's mediated. So I'm like, okay, that's the T cell. And then the humoral response is the B cells. Now, this is a little sciencey, but I think about bad humors and how that used to be referred to as like bad blood. So that's how I remember the B cells. I don't know if you're going to remember that, but try to think of a way to remember cell-mediated is T, humoral is B. Okay, so we're going to talk about how it is that this cell-mediated and humoral response even start going, and what is that link that kind of brings the second line of defense to the third line of defense. And it really has to do with the phagocytes that we talked about yesterday, those cells that engulf. One particular phagocyte is called a macrophage. There's actually a bunch of different white blood cells, all that go about engulfing, and their general term is phagocytes. There's one specific type that actually engulfs the invader and then displays their antigens, and that's a macrophage. So the macrophage eats the invader and then displays the antigens. So I guess a good way or a good example to think about that would be like if you're hunting and you come across a bear. If you take that bear and you kill it and you eat the bear and then you wear its fur as a coat, that would kind of be what the macrophage does. Sorry, that was a little violent there. But the macrophage engulfs the invader, and then it takes all of the antigens that were on the outside of that invader, and then it displays it all the way around. And again, remembering that antigens are unique proteins on invader cells. The antigen is basically the flag that says, hey, I'm Ebola. Hey, I'm COVID-19. Hey, I'm E. coli. So the macrophage wears these antigens, and then it needs help to figure out if this antigen is a good thing or a bad thing. Remember how we talked about the fact that there were antigens on everything that comes into our body. So the macrophage's job is just to eat things that are foreign, and it doesn't do a whole lot of thinking, so it needs help. So the macrophage takes antigens to a cell called the helper T cell, or a CD4 cell. So the macrophage presents its antigens much like one would present a coat. And it says, hey, look at these antigens. Do you recognize these antigens? Are these a pathogen that's going to attack us? Did I do a good job? Did I destroy something that was going to hurt us? And the helper T's job is to assess the situation and see, oh yeah, that is a terrible pathogen that could kill us. We need to mount an immune response. Or it says, no, you just ate some steak. Good job, I guess. So again, the macrophage takes it to helper T so that helper T has a final check to decide, is this something that is worth fighting? If the helper T decides that the antigens do represent a pathogen that is worth fighting, then what the helper T does is it binds to the macrophage and it releases a chemical called interleukins. Think of the interleukins as a chemical messenger that calls T and B cells to help. Cells can't actually talk to each other, so they have to send out chemical messengers. We learned this when we were talking about chemistry. So the interleukins are one of those chemical messengers. Essentially, they're kind of like the fire alarm. The job of the interleukins is to say, once these chemical messengers get spread throughout the entire body, the whole body knows that there is a pathogen that is trying to invade this body and it is dangerous and we need to fight it off. So interleukins are a key part to sort of alerting the whole body that we need to start mobilizing the third line of defense. Here's a picture kind of to represent that. So what you have here is you have the macrophage right here displaying the antigens. So you've got the macrophage displaying the antigens. Here's the macrophage. And it's got the little antigens on the outside of it like a little coat. See that there? And then what's happening is it's presenting its antigens to the helper T, and then the helper T is going to be activated, and that's going to release interleukins, right? Remembering that the interleukins are that call for help. So it's going to release the interleukins, which will tell the whole body to be on alert. Now, it's really important that interleukins are released. Interleukins have to be present for this next step to occur. In order for the T cells and the B cells to be activated, interleukins actually have to be flowing throughout the body. So the first step really is, in that link to from the second line of defense to the specific immune system, is really macrophage taking its antigen to helper T both sort of holding hands together, if you will, and calling for help by releasing interleukins. Once interleukins are present, then we can move on to the cell-mediated and the humoral responses. Now that we have interleukins coursing throughout the body, we can start initiating the cell-mediated response and the humoral response. Both of these can happen at the same time. One can happen before the other. It doesn't really matter the order. It kind of just depends on what happens in your body. But both are necessary in order to make the pathogen go away. So the first step is the immune system needs to find the correct cytotoxic T cell. A cytotoxic T used to be called killer Ts. They're also called CD8 cells. You might hear any of those terms, so I thought I'd just throw that out there. So they need to find the correct cytotoxic T cell out of millions of cells. So there are millions of different cytotoxic Ts that can actually take out different pathogens. Again, remember, each one is designed for a specific pathogen that you might encounter sometime in your life. So this is really hard. Your body has to find them. How does your body do it? The macrophage that's wearing that beautiful antigen coat is called an antigen presenting cell. So what it's going to do is it's going to actually take all of the antigens that it's wearing, and it's going to try to present them not only to helper Ts, but it's going to present them to a cytotoxic T cell. So it's going to go and any cytotoxic T that it sees, it's going to show it the antigen and say, hey, do you know this guy? Do you know this one? Do you know this one? And it's going to do this going across lots of different cells, millions of cells, until it finds the exact one cytotoxic T cell that can fight off the pathogen that relates to the antigen. So how does your body do this? Well, you start off with one cytotoxic T cell, or you start off with like one antigen presenting cell that has eaten a pathogen, but then as a virus, let's say, starts accumulating in your body, you get more and more macrophages that eat it until you have thousands upon thousands of macrophages that are all trying to find the right cytotoxic T at once. The other thing that your body does, which is kind of ingenious, is it sets up like a stop check system. Like if you've ever felt sick and you felt your neck swollen or underarms, those are major parts of your lymphatic system and your lymph nodes. Macrophages are part of your white blood cells, which live in your lymphatic system and travel in your lymphatic system. So what your body actually does is sets a trap in your armpits and on your neck in order to make all of the cytotoxic T cells that are floating around just kind of stop and check to the various macrophages that are going around with antigens to see if they recognize them. So long story short, it's a really hard journey to find the right one cell, but once it's found, it's cloned. And then once it's cloned, the job of the cytotoxic T cell, or the CD8 cell, is to find infected body cells and inject toxin into them, causing the cells to rupture and explode. So what the cytotoxic T is looking for is any cell in your body that has any sort of virus inside of it. And then it's going to, like a precision tool, take out those specific infected cells. This is a key component. So we need the cell-mediated response to take out the cells that are in the process of making new viruses. Because once a virus comes into your cell, the virus is going to take it over and it's going to turn that cell into a virus-making factory. So the job of the cytotoxic T is to stop those factories from making new viruses by taking out the factories. So here's a picture of the cell-mediated immunity response. So you have the macrophage down here if you're looking at number 1, and the macrophage is trying to eat an invader. So the invader is this the spiky little virus thing there, and its job is to try to eat that, right? So here's that little spiky thing is some sort of a virus, and the macrophage is going to engulf it, and then here we have the antigens being displayed on the edge of the macrophage. So it finds the helper T, and then we're releasing all the interleukins. Okay, so once we've done that, then it's going to go to step 2, where it needs to find the exact one where the macrophage is going to start presenting things until it finds the one cytotoxic T that can sort of destroy the pathogen. Once it finds the one cell, it starts cloning itself, and then in 4 here, you see the cytotoxic T. This would be like in 4, you see an infected cell, and the cytotoxic T just sort of injects toxins into it, and the whole thing explodes. Explode. So when that cell ruptures, if you can see all the little spikes on the outside of it, that's antigen that would be making new viruses. Now I've gotten rid of a virus factory. And then we're going to talk about this in a little bit, but then your body keeps a memory of that virus with a memory T cell. Okay, let's move on to the humoral response. In the humoral response, again, it's very much like the cell mediated. We have these antigen presenting cells that are going to try to find the one specific B cell that knows what this virus is. What happens is the B cell finds the B cell and it starts cloning that B cell. The B cell then turns into plasma cells, sometimes memory B cells as well, but we're more concerned with the plasma cells. The plasma cells make these things called antibodies. Now, we've talked about antibodies before with blood typing, but antibodies are really an important key to fighting a virus or any pathogen. Here's a picture to describe kind of how the humoral immunity starts. So again, we have this spiky little virus over here. We have the helper T, so we have like some sort of thing that has recognized it. Again, here's the little spiky virus. So what the spiky virus does in other antigen presenting cells is it finds the one, like it docks with helper T or some other antigen presenting cell that knows what the antigens are. In step 3, we found the exact one B cell that we need and it starts cloning. Then in step 4, we have the B cells are becoming plasma cells. These plasma cells make antibodies, and the antibodies are these little Y-shaped things. And their job is actually to cover free-floating viruses and make it so that they can't infect other cells. And again, like I said, we're going to get to at the end, then they keep a memory. So here's another picture just kind of showing you about the humoral response. Notice that each B cell is very specific to one antigen. So it's not the blue one, it's not the green one, it's not the purple one, it's the red one. And so that antigen that can dock with that B cell will allow that B cell to clone, make plasma cells, and then make antibodies. So how does an antibody work? It's a special protein that can attach to the antigens. And again, think of the antibodies as markers that help identify invaders for a cleanup crew. The other thing is if there is some sort of a virus that is infected with, or not infected with, but covered with antibodies, there's no way that that virus can enter other cells. So here's a really bad picture for you. So here's my little virus with all its little spikes on the outside, and these spikes are antigens, right? So if I have my antigens here, as the little spikes, then what happens is antibodies will come and attach to those antigens. Okay, a whole job of a virus is to trick a cell into thinking that it's a friend, and then the cell lets it in. Whee! Okay, when a virus is covered with antibodies, our bodies are like, hey, you're not a friend, you're an enemy. You're covered in antibodies, you're not coming in, and that prevents the virus from infecting other cells. Okay, so like I said, I was going to come back to the last step, which is keeping a memory of the invader. When the invader is eliminated, we keep memories. Memory B and memory T cells will remain in the body for the rest of our lives. If our body ever sees that same antigen again, aka that same exact pathogen, it will immediately make antibodies, immediately release interleukins, and it can fight off an infection way faster than it ever has before. Now it has to see that same exact antigen again. Viruses, bacteria are super good at mutating and changing themselves ever so much, like, for instance, putting on a fake mustache, and then your body doesn't quite recognize who they are anymore. So as long as it's the same antigen, your body can immediately mount a. So here's a picture of that memory. So the memory B cells here will instantly, so here, this picture right here is trying to show memory B, if it ever encounters a virus again, it immediately starts cloning those plasma cells and making antibodies. And here, if memory T finds an infected cell, it immediately starts cloning. This is over here. Here's an infected cell. It immediately starts cloning and making more cytotoxic T cells, so that hopefully with the combination of antibodies taking out free-floating virus and cytotoxic T cells taking out cells that are infected with virus, those two things together can defeat any pathogen. Okay, this picture is definitely really important. I definitely want you to draw this. So what this is showing is really a couple of different things. This graph right here, I'm going to call graph number 1. So graph number 1 over here is showing you the amount of antibody or T cells. This just means immune response. So this is how good your body is fighting something off, immune response. And you have days over here. So if we look at graph number 1, you can see that this is, let's say this is the flu. Influenza A. When you first get exposed to the antigen, it takes your body a couple days to start ramping up this system. In fact, you don't get a full immune response until about day 5. That's because your body has to find that correct one cytotoxic T and that correct one B cell that can fight it off. So once it finds that, it mounts an immune response, your body starts fighting it, you start to feel better. But those first few days, you don't feel really good because you feel sick and your body isn't really fighting it off yet. Then towards the tail end, around day 6 or day 7, you might feel a little bit better because your body has started fighting it off. Boom, you've had influenza A. Then, let's say you get that same virus a second time. You get influenza A again, and it's the exact same variety. Like, it hasn't mutated, it hasn't changed, it's the same type. Then we get to look at the little red graph over here. So this is the second exposure to the same antigen, and this is really important. So what happens on the second exposure to the same antigen is, notice how huge that immune response is right away. By day 2, it's already higher than it was at day 5 of the previous time that you had that illness. By day 4, the immune response is so great that what happens is you're actually, even though it's coursing through your body, you're actually not going to get sick from it at all. You're not going to feel sick. You're not going to feel tired. Your body just handles it. Okay? And this is because of the memory. Now, if, let's say, influenza A mutated and you get a new version, then you have to go through this whole immune response and memory one more time. But this is really important. This is a key part of how our immune system works, is we like to keep these memories so that we don't get sick from the same thing twice, or the same exact things twice. We're going to learn how viruses can mutate, how they mutate quickly to fool us, kind of like disguising themselves. And we're going to learn about how we have used this to our advantage with vaccines. Okay, that's it for the specific immune response. In a second, we're going to come back as a group, watch a video, and then I'm going to answer any questions that you have about the third line of defense.